Hello, today I want to talk about the accessory organs. And so remember, your accessory organs are going to be the organs that help with digestion, but they don't actually have foodstuffs entering them. So we're going to go through each of those and talk about how they contribute to the digestion of your food, both chemically and mechanically. So the first one are the salivary glands. They are this is the location of them. What their job is to bring saliva into the mouth. And saliva is important because it has both mucus and serous fluid that cause the mixing of the food with that and creating a bolus. A bolus is just simply a ball of food that has been partially digested because of the salivary amylase that has been secreted by those glands. The salivary amylase begins to digest starch, and starch is long chains of complex carbohydrates, and so it's going to chop those down into smaller bits. And it also dissolves those so that you can taste them as well. The pancreas, another accessory organ, produces enzymes that break down all categories of food, so fats or lipids, carbohydrates, and proteins. The enzymes from the pancreas are secreted into the duodenum or duodenum right here. And we also have alkaline fluid that comes in with those enzymes that's going to neutralize the acidic chyme coming from the stomach. So remember, the duodenum is the first segment of the small intestine. So right here, this would be connected to the stomach, to the pyloric sphincter. So that really acidic chyme is entering, and we don't want the acidity to burn up the walls of the intestine. And the intestine can't have that thick mucus lining like the like the stomach did, because the role of the intestine is to be absorbing nutrients. And so we need the foodstuffs to actually touch the walls of the small intestine. Hormones are also produced by the pancreas, insulin and glucagon, which insulin is going to lower blood sugar. Glucagon is going to raise blood sugar. So it's controlling the amount of sugar in your bloodstream. Here's the big picture. Here's the big picture of the liver. This is the gallbladder. It's connected by the cystic duct. It's coming in with the common hepatic duct. Hepatic means liver. Those join into the bile duct, and the bile duct is going to connect with the pancreatic duct and dump right in here. Remember, papillae means bump. The duodenal papillae is just where the ducts enter the duodenum. So the liver itself, frog, is the largest gland in the body. And when we do the liver is always amazing because it's very large when you look at it. When we do our dissection, you'll see that in the pig. It's located underneath the diaphragm. There are 4 lobes to it, and it's suspended by a ligament to the abdominal wall. So it's connected to the diaphragm and the abdominal wall by a ligament. And it connects the gallbladder, connected to the gallbladder by this common hepatic duct. So we get the connection of those. What is bile? Bile is produced by the cells of the liver, and it's going to leave the liver through the common hepatic duct. And what it is it contains salts and pigments, which come from the breakdown of hemoglobin. So remember, hemoglobin is the protein that's carrying oxygen in our red blood cells. It also contains cholesterol, phospholipids, and electrolytes. What it does with all of those ingredients is that it's able to emulsify fats. So what that means is that you have a big, large fat globule, and when it comes in contact with the bile salts, there's a hydrophilic side and a hydrophobic side to these salts. And what they do is they surround the fat. Remember that fat is hydrophobic. So you have a big chunk of fat, and the hydrophobic sides are going to connect to the sides of the bile salt that's also hydrophobic to try to get away from the water. And so by doing that, it does this process called emulsification, where it's just going to break them apart. So this is a type of mechanical digestion because we're not dissolving the fat. We're just breaking it into smaller droplets. The gallbladder works in conjunction with the liver. It's found on the bottom of the liver, the bottom side. And when we are not digesting, bile is going to be stored in the gallbladder. So it backs up into the cystic duct and is stored there. When digestion of fatty foods occurring, then it's released into the duodenum from the gallbladder. And people have experienced gallstones. That's crystallized cholesterol, which builds up. So the salts crystallize and the cholesterol builds up and can cause blockages, and that can be really not fun. All right, so that's this portion of what we were looking at. And now I want to present a little bit on the whole process of digestion. So let's go back to reminding you of what the steps are in digestion, what all the processes are. So ingestion is eating, propulsion is moving the food, peristalsis is how propulsion happens. It's the contractions when the smooth muscle, the circular and the longitudinal muscles work together to bring food down. Segmentation, I'm going to show you a visual of that in just a second. Segmentation is what happens in the small intestine. And so those are the ways that the food is going to move through. So I'll come back to this. This is a little crazy. So let's look at this first. So mechanical digestion means that it's just making smaller pieces. So the function of that is really to increase surface area of the food. So how this is achieved, we mix food with our tongue. We have that churning in the stomach that we've looked at. We have segmentation in the small intestine, which I'll show you in just a second. And again, the whole purpose is to prepare the food to be degraded by enzymes, which would be the chemical digestion. But this whole sentence basically means to increase the surface area that's going to be exposed to the enzymes. This is segmentation. So what segmentation is that we're in the small intestine now, and there's contractions that are going to move that food back and forth into little segments. So right here, there is not food here, and there is food here. Well, then when this contracts, it's going to mix this portion of food with this portion, and this portion with that portion. And so then we get more mixing. So it's just like a little bit of it goes here, and then we cut half of it off, half of it goes up, half of it goes down, and mixes with the stuff above it, and mixes with the stuff below it. So that's what's happening in the small intestine itself. So that was mechanical, but then food, once it's mechanically digested, then we can work on it with some chemical digestion. So remember, enzymes break polymers, long chains, into monomers, building blocks. And so what's going to happen is that we have different enzymes. Remember, enzymes are specific. So we have carbohydrates that are broken down into simple sugars. We have proteins that are broken down into amino acids, and fats are broken down into fatty acids and alcohols. And so different enzymes are going to work on the different food sources in different locations. So we're going to start with carbohydrates. The carbohydrates, just looking at this diagram. So it's looking at what foods come in, what enzymes work on it, and where that happens. So we have longer chains. Starches are the longest. Polysaccharides are starches, and they are going to be broken down into disaccharides by the enzymes in your mouth called salivary amylase. Then they're going to continue, and we're going to get more amylase, only this one's coming from the pancreas in the small intestine. And so that's going to break it down into even smaller bits. And then in the small intestine, we have brush border enzymes that are going to break down the specific disaccharides into monosaccharides. So monosaccharides are going to be the smallest, and we have different types of monosaccharides depending on what your source of carbohydrate was. But this is going to happen in the small intestine. So this is primarily coming from the pancreas when we dump this into the duodenum. So monosaccharides of glucose, galactose, and fructose are the only ones that can go across the capillary wall into the villi, and then they get transported from there. So oligo means different. That word oligosaccharide, that just means that it might be a few linked. So di means two. So we have multiple poly up here. And then when we are exposed to these, they break them down into smaller chains, and then finally into the ones that are monosaccharides, and those can go across for carbohydrate absorption. Okay, so now looking at protein, you'll notice here nothing is happening in the mouth. The very first place that protein is digested is in the stomach. And we talked about that pepsin is in the stomach, and it is converted from pepsinogen in the presence of hydrochloric acid. So when you have actual food in your stomach, the hydrochloric acid is going to be released. When the hydrochloric acid is released, then the pepsin becomes activated and breaks down that protein. So it's going to break it down into polypeptides, so that the protein itself is going to be a really large chain protein, but it's going to break it down into poly, so a little bit smaller. And then the pancreatic enzymes, there's trypsin, chymotrypsin, and carbopeptidase in the pancreas that are going to work when we're in the small intestine. So again, the duodenum of the small intestine, that is going to break it down into smaller polypeptides, and then your brush border enzymes are going to break it down even further. So remember that a monomer of a protein is an amino acid. So this is easy to remember, aminopeptidase. So it's going to break it down into amino acids, carboxypeptidase, and dipeptidase. So if you look for the peptidase, remember, enzymes end in ASE most of the time. Trypsin and chymotrypsin are exceptions, but ASE, and then you can kind of figure out what they're breaking down by what the names are. So polypeptide chain, remember that's a chain of amino acids. So you should be able to rationalize what those are. Amino acids are going to enter the capillary blood in the villi of the large intestine and be transported again to the liver from there. Fats, no mouth digestion, no stomach digestion. Here they're emulsified when they're entering, meaning that unemulsified, meaning that they're all in one big globule. And then in the small intestine, when we have bile coming from the liver, we're going to get them broken down into smaller and smaller globules. And then the pancreatic lipase, lipid ACE enzyme that breaks down lipids, is going to enter, and it's going to chemically break them down into monoglycerides. Mono means one, and glycerol and fatty acids. So fatty acids and monoglycerides are going to enter the lacteals of the villi, just like everybody else, and be transported to the systemic circulation through the lymph in the thoracic duct. So they're going to be absorbed into the capillary of the villi and transported to the liver. So basically it's transported just a little bit differently, but the end goal is the same. It's going to end up going to the liver. So that absorption piece, now that we've broken everything down, is that the end products are going to enter the blood or the lymph, and they're going to, in order for that to happen, it has to enter the mucosal cells to get there. And so the absorption piece is all in those, all in those lacteals with the microvilli, the villi and then microvilli, getting those enzymes to break down the food into small enough sections so then it can be absorbed right into those capillary beds in that lacteal. And then there's defecation. So here's that diagram that is a little bit wild, but it's trying to put it all together. So it's showing ingestion is eating, and then mechanical digestion, we're getting the chewing with our teeth, the churning in our stomach, the segmentation in the small intestine. And then in order to get it to go down, we need propulsion, which is going to be achieved by the swallowing with the oropharynx, and then peristalsis is going to be that circular muscle and longitudinal muscles working, and that's going to happen pretty much throughout. Peristalsis does. And then we start the chemical digestion. They should have had one fat globule or one globule up here talking about chemical, because the salivary glands produce salivary amylase, which is going to break down carbohydrates. But most of the chemical digestion is going to occur, some of the protein in the stomach, and then right here when the pancreas dumps in, that's gangbusters, where we're going to get a lot of it. We get absorption throughout the small intestine through those lacteals. Once we get to the large intestine, there isn't absorption anymore, except for we're reabsorbing water into the blood vessels from the leftover undigested materials. So I think that is where I want to stop today.